Started. Hello, everybody. Welcome to another episode on our Western Evolution, Biology Evolution Seminar Series. Again, this is an online series that we have developed to really help spread the knowledge and research on evolution. And today I have the pleasure to have here with me Dr. Marc Johnson from the University of Toronto, Mississauga. And his talk, as you can see, is titled Evolution of Life in the Urban Jungle. But before I pass it on to Dr. Marc, I would like to introduce him more formally. Dr. Marc Johnson is a professor at the University of Toronto, Mississauga, and the Canada Research Chair of Urban Environmental Science. He obtained his PhD from the University of Toronto in 2007, after which he conducted a NSERC postdoctoral fellowship at Duke University. And he was an assistant professor at North Carolina State University before returning to UFT in 2011. His research takes an interdisciplinary approach to address a broad array of problems in evolution and ecology, including co-evolutionary biology, eco-evolutionary dynamics, the evolution of sex, adaptation to climate change, and how urban environments affect the ecology and evolution of life around us. He has received several awards and fellowships, and he's presently an associate editor at Proceedings of the Royal Society of Biology, chair of the board of Support Our Science, which through he advocates for increased funding for graduate students and postdoctoral scholars in Canada. He is also director at the Riverwood Conservancy, co-founder of the Riverwood Young Naturalist Club, and the founding director of the Center for Urban Environments. Dr. Marc Johnson, thank you very much for being here with us today, and the floor is all yours.
Thank you so much. It's wonderful to be here with you online and virtual and spreading information about evolutionary biology. So yes, I'm going to tell you about evolution of life in the urban jungle today. And before I do that, I just want everyone to know where I'm coming from. I'm currently on the, if we can get this going, currently on the shores of the Credit River, the Mississauga in Anishinaabe, which is the traditional territory of the Huron-Wendat for thousands of years, and more recently the Anishinaabe from the Mississaugas of the Credit First Nation. And feel very fortunate to be working on this land today and starting to understand it and through the lens of our research. And it's at the University of Toronto, Mississauga that I run the Evo Eco Lab, which we established at North Carolina State University 14 years ago. And we study the intersection of ecology and evolutionary biology, trying to understand the dynamic interplay between ecological processes and patterns and how they influence evolutionary processes and patterns and vice versa. But we do this not just by looking at ecological and evolutionary questions and using those techniques. We also integrate it with genetics and chemistry, as well as genomics and lots of statistics. And by incorporating these four areas of research, we're able to answer a number of questions at the intersection of these fields, including plant-herbivore coevolution, plant-soil feedbacks, eco-evolutionary dynamics, as you're mentioning, evolution on islands, and the evolution of sex. But today, I'm not going to be talking about any of these research areas, although they're still active areas of investigation in my lab. Today, I'm going to be trying to answer one very general question with you, and that is, how do urban environments affect the evolution of life? This is a topic that I've only become interested in relatively recently, probably about the last 8 to 9 years. And I became interested in this because I began to realize, like many people watching this realized long before I did, that we're living in a fundamentally different time on our planet. Geologists are starting to recognize that we're living in what they're calling a new geological epoch, the Anthropocene, where humans are having a decisive influence on the state, dynamics, and future of the Earth that is fundamentally different than before advanced developments and industrialization. At the same time, social scientists have also started to say that we're living in a century that is fundamentally different than previous centuries, and they're calling this the century of the city. And they're calling it the century of the city because as of 2008, we passed a critical threshold that we will probably never go back from. Before 2008, most humans lived outside of cities, in rural areas mostly, but also natural areas. But as of 2008 was the year in which we passed the threshold where more than 50% of humans were living in urban areas, and less than 50% were living in those rural and natural areas. In Canada, we passed this threshold a long time ago. Today, 82% of Canadians live in cities or some type of urban development, including towns and villages. And when we look across the world, we see that this is truly a global phenomenon, and that there are thousands of cities across all inhabited continents of this world. So this isn't something that's particular to Canada. It's really going on all around the world today. And these urban areas make up relatively small percentage of the terrestrial surface of Earth, 0.5% of the Earth's land surface, but it is the fastest expanding ecosystem on Earth. Urban areas grew on average by a little under 10,000 square kilometers per year between 1985 and 2015, and the rate of this change is continuing to escalate. And so we are rapidly urbanizing the Earth around us. And I think this is an important question. And so I've been trying to understand how we're changing the environment around us and the ecological and evolutionary consequences of this. So if you look at urbanization, it leads to a number of predictable environmental changes, which we can think of as being a syndrome, an urbanization environmental syndrome. So there's numerous aspects of the abiotic environment, the physical environment, from water stress to temperature to salinity to pollution, that predictably change as you go from natural environments to rural environments to the suburbs and downtown areas. At the same time, there's a number of biotic factors that change. This is the living environment. So things like mutualists like pollinators, antagonists like parasites, as well as the habitat composition are influenced by urbanization. So for example, if you go to the city that I live within, which is the Greater Toronto Area, if we go from downtown Toronto in this area around here, and we follow this little track going west or north or east, we're going to see predictable environmental changes as we go along that transect from the downtown area out to the rural and natural areas around Toronto. So for example, let me just show you some data that was actually taken from each of these sampling points along here. So I'm going to show you a lot of figures like this. So let me orient you to this one, and then they'll be easier for you to interpret after this one. So as we go from on the x-axis here, we go from the left to the right. This is increasing distance from the urban center, where the center of the city is denoted as 0. The furthest sampling point is denoted as 1, and 0.5 is about the threshold at which we go from suburbs to more rural areas, or some people call them ex-urban areas, but much lower density and farmland around. We standardize them from 0 to 1 just because different cities, when we start looking at different cities, can be of different sizes, and so it's helpful to have them on the same scale so we can compare apples to apples. On the y-axis here, we have the response variable, in this case, percent impervious surface. This is simply the amount of surface area around that sampling point, in this case within 250 meters, that is made up of concrete, asphalt, or buildings, so it's impervious to water penetrating in there. And each one of these dots here is a sampling point, so a location in space along one of those transects. And those transects were selected so that there's 1 kilometer in between the sampling points. And we're fitting here the line of best fit from a linear curve. So what you can see is that percent impervious surface is very high in the downtown areas, and then linearly decreases, or more or less linearly decreases, as we go further from the city, from urban areas to rural areas. At the same time, we see an increase in the amount of vegetation measured as NDVI, the Normalized Difference Vegetation Index. It starts very low in the city center and increases as we go to rural areas. And we see a decrease in surface temperature where it's relatively high in urban areas and decreases as we go out to rural areas. And this is that classic urban heat island. So these urban environmental changes, this urban environmental syndrome, is a well-known phenomenon. We've known about it for decades. And so what I'm showing you is not new. But what we haven't understood is how these environmental changes associated with urbanization may influence not just the ecology of systems, but may influence evolutionary change over short spatial scales and short time scales. And so given this context, I'd like to try and answer the question, how do urban environments affect the evolution of life? I think this is a really exciting question, both from a fundamental science perspective and from an applied science perspective. From a fundamental perspective, you can think of urbanization as the best replicated, largest scale experimental evolution study of all time. There are literally thousands of cities around the world in which we have frequently changed the same environmental factors. We've had increased impervious surface, more buildings, more pollution, more roads, more people. How do these consistent environmental changes alter evolution? We also have some features of cities that can be different from one another. Some may be in tropical regions, some may be in temperate regions. So also, how do these differences between those cities influence evolution? At the same time, although this is a very new topic in some ways, urban evolution can be thought of as an old topic. This may be surprising when I first say it, but if you think of the very first examples of evolution in action in nature that we learn about, it is things like the peppered moth, studied by Kettlewell in the 1950s. This rapid evolutionary change was caused by industrial pollution associated with cities and how it changed natural selection on this moth species, increasing selection for the melanic form on the left here, but then when pollution was abated, led to increased selection or selection for in favor of the lighter wild type morph, leading to a decrease in the melanic morph. So we have, even though it hasn't been a topic that has been studied a lot until recently, some of the earliest examples were you could think of as being urban evolution. But in a lot of ways, it's undeniable that urban evolution is also a new topic. So this was the first review, kind of comprehensive review of this was done by Jason Munchie South and myself in a paper in Science in 2017, going on 6 years now. And what we attempted to do was to do a comprehensive review of all the literature that has been published on the effects of urbanization on evolution. We didn't think that we were going to find that much because we thought there was very few studies, and there was relatively few, but we were surprised to find that there had been 192 studies on 134 species represented by these species across all of these different places around the world. A lot of times these studies were not pitched in a context of studying the impacts of urbanization on evolution, but they were done in a geographic location where we could actually get some insight about that. And at that time, over 50% of these studies were from the past 5 years. Now, since that time, we've seen an even greater than doubling of the number of studies. This has become a very hot topic in evolutionary biology. And so our knowledge is rapidly expanding. What we discovered in the course of this review is that cities can affect evolution in 4 main ways, where the development of cities can affect mutation of individuals, both somatic mutations and also germline mutations, genetic drift, which is defined as stochastic changes in allele frequencies within a population through time, gene flow, so that's the dispersal of individuals and their alleles combined with reproduction, leading to the movement of and changes in allele frequencies from one population to another population. And fourth, natural selection. And I have a question mark here beside natural selection because at the time we wrote this review, and it's still very much true today, we knew very little about how urbanization influenced natural selection, often also called Darwinian selection. And so this is something that, because we knew so little about it, that we've been investigating quite intensely for the last 6 or 7 years. And we've been doing this in a number of systems, including interactions between Darwin's finches, uh, and the foods that they eat, as well as looking at different insects on plants and the plants that they're feeding on, milkweeds and milkweed insects, as well as this plant here, spotted touch-me-not. But the plant I'm going to talk to you about today is white clover, Trifolium repens, where you can see its leaves here in the top left, its flowers in the top right, and it forms these large mats that many of you will be familiar with because they are found pretty much in every city around the world. So they're extremely common throughout the world. And using this system, we want to try and address this question: can populations adapt to urban environments? And by adaptation, I'm not referring to physiological changes, but specifically evolution by natural selection, the original definition of adaptation. We use white clover initially because it has a very interesting genetic polymorphism called cyanogenesis. Cyanogenesis is a potent anti-herbivore defense. If we go out onto my lawn right now, what we're going to find is that some of those plants are going to not produce any hydrogen cyanide, so they're going to be acyanogenic. And then other plants are going to be cyanogenic. They're going to produce hydrogen cyanide in their leaves. And this is a very, very potent defense. If there's hydrogen cyanide, if you ingest hydrogen cyanide, it attaches to your cytochrome oxidase in your mitochondrion and shuts down the electron transport chain, so you can't produce any ATP. And you need ATP to fuel the energy that your cells use to perform pretty much all of their functions. This hydrogen cyanide in the leaves of white clover is released following tissue damage. So if an insect comes along and eats the leaf, it releases this hydrogen cyanide. If the tissue freezes, it can release this hydrogen cyanide. And this presence or absence of hydrogen cyanide is controlled by two loci, and each of those loci have two alleles. So the first locus is CYP79D15 and two other genes that are in tight linkage with this gene. So it's not entirely clear if it's CYP79D15, but they're all involved in biosynthesis of cyanogenic glycosides, specifically linamarin and latostrolin. Plants need to have at least one functional copy of CYP79D15, which I'm going to denote big AC, in order to produce cyanogenic glycosides, linamarin and latostrolin. If they have a gene deletion, which is very common, then they are, and if that gene deletion is homozygous, then they don't produce any cyanogenic glycosides. So big AC is dominant in its expression. They also need to have one functional copy of the LI gene, which encodes for the enzyme linamarase, which cleaves the glycoside to make hydrogen cyanide. And again, they need to have, I'll denote this big LI for the functional copy, which also has dominant expression. And again, there is a recessive copy in which there's a gene deletion, and if they're homozygous for the gene deletion, they don't produce any linamarase. A plant needs at least one copy of big AC and one copy of big LI in order to produce hydrogen cyanide. Well, we were not the ones to discover this, far from it. The first work on this was actually done in the early 1900s, and then as an evolutionary model was developed by Day. And what Day discovered was that as you go from the south and coastal regions of Europe, where this plant is native, to the north and eastern regions, so moving away from the Atlantic coast, you have a decreasing frequency of big AC and big LI, meaning that you have more acyanogenic plants. So in the very south of Europe, all those plants in those populations are cyanogenic. In the very north and east of Europe, all of those plants are acyanogenic. And this decline in cyanogenesis follows a temperature gradient, which is shown by these isotherms, which are these squiggly lines here showing the increasing temperature in winter as you go from left to right. And as a result of this, although hydrogen cyanide is involved in defense, Day hypothesized that it was really the cost of the defense when tissues are frozen that drives the evolution of this cline. Because if the tissue freezes, he hypothesized that the plants could be poisoning themselves by releasing hydrogen cyanide that inhibits the plant's own machinery, molecular machinery. Now, there's since been a lot of debate about whether it really is freezing or not. I'm not going to get into that today, although I'm happy to answer questions because we've been working on that, as well as some of our close collaborators have worked on that in the last 10 years. But I was interested in not just whether this is playing out, how this is playing out at continental scales, which is an interesting question, but whether these same type of gradients in temperature could be leading to, and potentially also herbivores, could be leading to the evolution of cyanogenesis across not an entire continent, but across a small or across a city. Because as you go from downtown regions of a city like Toronto here to the outlying regions of Toronto, you also have a change in temperature, which you can see from this map showing surface temperature across Toronto. In the center here, we have downtown Toronto, and you can see it's quite, the warmer temperatures indicate hotter surface temperatures, as indicated by this little legend at the bottom right. As you go to the suburbs where there's very few trees and very dense housing, it gets really, really hot on a warm summer day. But by the time you get to the rural areas and natural areas outside of Toronto, the surface temperatures can be very, very cool. And so we get the same type of temperature gradient over a fine spatial scale as we do across an entire continent. And just for some sense, you know, the surface temperature and particularly the air temperatures, you go from downtown Toronto to the rural areas would be like going from here down to South Carolina. That's how big of a difference it can be on a given day. So our hypothesis was that cyanogenesis will evolve in response to urban temperature gradients if there's similar selective pressures and similar evolutionary processes as Hunter was seeing at these continental scales. And if this is true, if we have the same type of continental scale evolution playing out across a city, then we'd predict that urbanization will cause a decline in cyanogenesis, that hydrogen cyanide. That's our first prediction. And second, that cyanogenic genotypes will be more common in cities than in rural areas, just like Hunter found that cyanogenic genotypes were more common in hotter areas of Europe. We predicted that we'd find more cyanogenic genotypes in downtown Toronto. So to test these predictions and hypothesis, I collaborated with a former master's student in my lab, Ken Thompson, who's now doing a postdoc. He's since done a PhD and is now doing a postdoc at Stanford University. And what we did together was we sampled 3 transects across Toronto. So we went north. Let's back up here, sorry. North, east, and west. Each transect was 50 kilometers. And all we'd do is we'd start at the end of one transect, and then we'd just set the odometer, get out after a kilometer. There'd always be plants there pretty much. We'd sample 20 plants, label the bag, put it in a cooler, get back in the car, go another kilometer. We just do that all day for 3 days. And then when we got back to the lab, Ken assay the presence or absence of whether the plants were producing hydrogen cyanide, and then whether or not the plants had the big AC or little AC copy of the gene, or the big AC, big Li or little Li copy of the gene. What we found, this started as a side project. We didn't know what we'd find at all, although we had these predictions. What we found was really quite stunning and in a lot of ways surprising. As you went from downtown Toronto, again, that's on the far left here. So this is distance from the city center again. 0 indicates downtown, to the furthest rural populations, which is kilometer 50, we saw a clear increase in the proportion of plants within a population that produce hydrogen cyanide. So each dot here is a separate population, and this is the frequency of, out of 20 plants, of plants that are producing hydrogen cyanide. And amazingly, this explained 43% of the total variation in the frequency of cyanogenic plants within a population. So this was from the west transect. We saw the exact same result on the northern transect and on the eastern transect. And the slopes of these lines are statistically equivalent to one another. So this is basically the same result on all of those clines. So if we go back to our predictions, we found that urbanization does cause a decline in cyanogenesis, absolutely. But we did not find that cyanogenic genotypes were more common in cities. In fact, we found the exact opposite, which was very surprising to us at the time. So to try and follow up on this, we did a number of experiments to try and elucidate what was going on. It still was rather unclear. We sampled a number of cities across that of different sizes to see how that could be influencing things. This was in collaboration with an undergraduate student, Cindy Prashad, and a visiting master's student, Melanie Lavignon. And again, we found similar results where they're evolving to have increased frequency of cyanogenic plants and genotypes in rural areas, fewer hydrogen cyanide producing plants in urban areas. And we also did this along latitudinal gradients. This was the work of former PhD student, now a postdoc at Berkeley, James Santangelo, where he found that these declines get even stronger as you go further south, which was also a little surprising to us. So in trying to come back to this question, can populations adapt to urban environments? So far, the results are consistent with adaptation to urban environments in that we're seeing responses to the environment, and we're seeing these evolve over and over and over again in different cities in different environmental contexts. And when you see parallel evolution like this, it is often seen as some of the strongest evidence of adaptive evolution. But let's hold on to that thought. So at this point, we'd sampled a lot of cities in and around Ontario and in Eastern North America. And we had really established very clearly a lot of the methods on how to do this and made it pretty foolproof on how to sample plants and assay whether or not they produce hydrogen cyanide, and therefore understand what evolution of genotypic frequencies and gene frequencies in these populations. So we saw this as an ideal system in which we could understand whether urban populations adapt to urban environments in similar ways throughout the world, not just in the small number of cities that we've looked at, but maybe potentially throughout the entire world. And we saw white clover as one of the very few systems where you could study responses to urbanization at a global scale, because although white clover is native to Europe and Asia, particularly the Middle East, it has been introduced as forage and as a fertilizing plant throughout the world: North America, South America, Africa, particularly Southern Africa, Australia, New Zealand, and Asia. And so we saw this as a way to go global with white clover, trying to understand whether urbanization drives similar types of evolutionary processes and patterns in response to urban development in cities throughout the world, regardless of whether you're in Tokyo or Toronto, or Melbourne versus Munich. And so we launched what we have called the Global Urban Evolution Project a number of years ago, which this is the website, and you can visit it and learn more about the project. And we shorten this just to so we don't have to chew on that entire mouthful when we say it, to the GLUE project. And with the GLUE project, we're trying to answer 4 specific questions. And that's what I'm going to tell you about in the rest of this talk. First of all, does urbanization lead to convergent environmental change throughout the world? We see this as an important, almost necessary condition in order for urbanization to lead to parallel evolution. Second, does this convergent environmental change cause convergent or parallel evolution in an ecologically important trait, hydrogen cyanide? Third, what environmental variables drive convergent evolution to cities on a global scale? So really trying to get at what are the agents of natural selection that could be causing this environmental change? And finally, do other regions of the genome adapt to urban environments in parallel? So we've been, and I will for most of this talk, really focus in on hydrogen cyanide, but of course there are thousands of genes, tens of thousands of genes that could be responding. To launch this GLUE project, which was very ambitious, admittedly, we harness not only our own collaborators, I have a network of collaborators around the world, but we harness the power of social media. And so after contacting a few collaborators that I was very close to and asking, hey, would you be interested in working with us on this? I was amazed that everyone I contacted said, absolutely, this sounds really exciting. I'd love to be a part of this. And so we knew that that people were generally supportive of it. And then when we launched it on social media with this tweet on January 18th in 2018, I wrote, we're seeking collaborators to participate in the Global Urban Evolution GLUE project, a global study that seeks to understand whether urbanization drives parallel evolution in cities throughout the world. And we gave a link where people could sign up. And it didn't quite go viral, but it got very popular very quickly. So that, you know, lots of people started contacting us about it. Lots of people started inquiring. Within 8 days, we were already at 129 cities as shown in this map. And our original goal was to try and get to 50 cities. And then very quickly, when we blew past that, we said, okay, let's try for 100 cities. That was in the first 2 or 3 days. And then after a week, we said, let's try for 200 cities. We got up to 187, but for a variety of reasons, it dropped back. And so we still got a lot of cities, 160 in total. And in total, we had 287 collaborators sign up to participate in this study with us. So it was really a fundamentally different way of doing science, and the largest scale study of evolutionary biology ever, and the largest collaborative study in evolutionary biology that I'm familiar with. So in total, as I mentioned, these are the cities that we sampled across the world. Involved 160 cities in 26 countries from all inhabited continents. Together, all of these collaborators sampled over 110,000 plants from over 6,000 populations. So a very, very large study. Most of those were in North America and Europe, but we did have representation in Asia, South America, Africa, Australia, New Zealand as well. When people were sampling, they started sending us pictures and posting them on social media of their sampling and the animals that they were coming across, the plants that they were finding, the assays that they were doing, and the hard work that they were doing, as well as the kids that they got involved in this work. And not only the kids, but also the pets that they involved in this large collaboration. People were clearly having a lot of fun while they were doing this, and liked showing us on social media and other ways of how they were participating in the research. And so it became this very enjoyable collaborative and collective effort where we were all bound by the exact same methodology, the exact same sampling protocols, doing the exact same study in cities right across the world. To give you a sense of how this was done, I'll just show you London, Ontario, which is kind of where this online seminar is based. London, Ontario is a medium-sized city in Ontario. I think, if I recall correctly, there's somewhere around 400,000 people in London. And the outskirts of the city on this satellite image are shown in red here. And we sampled every population shown in these purple dots right here. See if you can still, there we go. By these purple dots that you see starting in the very center of the city and then following this transect to the northeast of the city. We sampled 20 plants from each one of those locations. I think there was a total of 40 populations in London, Ontario. But then from each of these, we also sampled each of these locations. We used satellite imagery and remote sensing to estimate percent impervious surface, summer temperature, and the amount of vegetation around each sampling location, as well as a number of physical features such as aridity, evapotranspiration, snow depth in winter, winter vegetation, and winter temperature. So in total, we had these 8 variables plus elevation, so 9 environmental variables for each location. And then we tried to relate how these environmental variables were associated with changes in the frequency of HCN within populations. So this is the actual curve for London, Ontario. So you can see it's curvilinear in this case. The frequency of hydrogen cyanide within populations goes up and then falls. So it's at a high point out in the suburbs of London. So let's go to this first question. Does urbanization lead to convergent environmental change throughout the world? To answer this question, we used multivariate statistics. So we're trying to take all of those environmental variables and then decompose them into a smaller number of variables. And we do that using principal components analysis. So we have 2 PC axes. PC1 on the x-axis here, PC2 on the y-axis. And together, these two environmental variables explain 65% of the total environmental variation. Each point here represents a city and the city's environment. So in green, we have the rural environment, and that we'll point to this one here, the rural environment. And then that is connected to the same city and its urban environment. And what you can see is that there's this consistent shift in the environment as you go from rural habitats in green here to urban habitats in orange here. And this difference, this multivariate difference, was in fact highly significantly different, such that it was often the case that two cities would have a more similar urban environment to one another than either city did to its nearby surrounding rural environment. So in the case of Tokyo and Toronto, their urban environments are more similar to one another for these environmental variables than either one is to the surrounding outlying rural areas, even though they're on different parts of the globe, which is really quite stunning. And we can see which environmental variables were associated with this change. It was largely surface temperature in summer, the amount of impervious surface, the amount of vegetation, and the amount of and the aridity. These other environmental variables, the snow index, winter temperature, winter vegetation, evapotranspiration, and elevation, they were often associated with changes in urbanization, but not in a consistent way. It was cities often differed in how those environmental variables changed for these other variables that are going perpendicular to the main axis. But these four consistently changed in pretty much every city. So given that we saw convergent environmental change, our second question was, does convergent environmental change cause convergent evolution in an ecologically important trait? So if we look at three example cities here, we can see a city like Temuco, Chile, down in southwest South America. We see no change in the frequency of hydrogen cyanide. It's relatively high frequency in Temuco, and it doesn't change as you go from urban to rural areas. Then we see other cities such as Münster, Germany, where the frequency of hydrogen cyanide is very low in the urban areas on the far left here and increases quite dramatically as you go to surrounding rural areas, much like we see in Toronto. And then you find some cities like Freehold, New Jersey, where we get the opposite result, where the frequency of hydrogen cyanide is highest in the city and decreases as you go to rural areas. Well, if we look at all 160 cities, we can plot the slopes of this change in a frequency histogram. So now we have the slope of change in hydrogen cyanide with distance from the city center on the x-axis here, and the number of cities that have different slopes. So that this little dotted line that I'm pointing to right here in the middle is where the slope is 0. The overall average across all cities was positive, shown by this dashed line, and the main effect of distance from the city center across all 160 cities was highly significant, but there was variation among cities. All the cities in blue showed, if you went kind of city by city and did these analyses, showed significant declines in hydrogen cyanide frequency, and in total 39% of all declines were positive. All those in red, which represented a minority of declines, showed a negative relationship, so that they had highest HCN in the city, like Freehold, New Jersey. And then all the ones in gray were ones that didn't show a significant change in hydrogen cyanide frequency within populations in response to urbanization. In total, almost half of the cities showed a significant change in the frequency of hydrogen cyanide with increasing distance from the urban center. And if you want to look at all 160 of those declines, at least looking at the lines of best fit, you can see that here, and that most of the ones that are significant in blue are going up here, and then the minority in red are going down, and then the rest are in gray. So given that we see these repeated declines, what evolutionary processes explain urban-rural declines in cyanogenesis? And I think it's particularly powerful that we have variation in the direction of these declines and the strength of these declines, because that's the type of variation we need in which to be able to tease out what are the environmental drivers of this. So first of all, parallel evolution, like we see in part from this study, is evidence for adaptation, or often seen as evidence for adaptation. But I want to caution in a very quick interpretation of that and say not so fast, because you can actually get parallel evolution, like the 39% of cities that show a positive decline, due to genetic drift alone. And that may seem surprising to the audience, not thinking that you can get repeated declines in that way. But we published a study in 2018. This was a study that was led by a PhD student that was co-supervised by Rob Ness, shown on the right here, and James Santangelo. And we did a number of simulations together using the genetics of the hydrogen cyanide system, which has these two epistatically interacting genes, which have dominant expression. And if you have a homozygous recessive for either locus, the plants are acynogenic. So if they're little ac, they're acynogenic. Or if they're little li, they're acynogenic. Because of this epistatic interaction, genetic drift is more likely to lead to the loss of hydrogen cyanide than its gain. So in thinking about what evolutionary processes can explain urban and rural declines in cyanogenesis, neutral evolution due to genetic drift and in combination with population bottlenecks or reduced population size can lead to consistent, repeated directional changes from cyanogenic populations to acynogenic populations. At the same time, a second process that can lead to urban and rural declines is variation in gene flow between populations, where maybe you're more likely to get declines in cyanogenesis if you have restricted gene flow between urban and rural areas. And finally, of course, natural selection can lead to changes along these gradients. So if we think about the first process, neutral processes, if we have a population that starts as having both cyanogenic and acynogenic plants, and if it goes through a bottleneck, because just fixing the recessive alleles at one locus leads to the loss of cyanogenesis, population bottlenecks will frequently lead to a reduction in the frequency of cyanogenesis within populations and potentially the complete loss of cyanogenesis. So if we have a case where white clover is invading cities from rural areas, like we actually think that this is a realistic scenario in many cities, if those rural populations are polymorphic, and if they start to colonize urban areas through kind of this stepping stone process, then genetic drift itself, as a result of these bottlenecks, could lead to the loss of cyanogenesis in urban populations. We can't study this process directly, unfortunately. But if there is this kind of dispersal from rural areas into urban areas, this is expected to lead to a predictable change in genome-wide genetic diversity, such that if you have repeated bottlenecks as you invade a new environment, this should lead to a loss of genetic diversity. So we can look across the entire genome and ask, do we see that predicted reduction in genetic diversity if neutral processes are leading to these repeated evolution of declines? Well, when we look across all of our populations, we were able to sample plants from 26 cities for whole genome sequencing. And in total from these 26 cities, we initially sequenced 520 genomes at low coverage, 1.25x, which produces a lot of data and allows us to estimate signatures of genetic diversity or genetic diversity directly from the site frequency spectrum. The prediction is that if urbanization leads to reduced population sizes, so these population bottlenecks that can lead to these types of evolutionary declines due to neutral processes, then we expect to see lower genetic diversity in urban areas than in rural populations. And that if we look at the slope of the decline of HCN across different cities, as a function of the difference in genetic diversity between rural and urban habitats, we'd expect a negative relationship. So this is the prediction. This isn't the real data. Now, if I now show you the actual data from these 26 cities, And these 26 cities represented variation in the strength of clines and the direction of clines and all of that. We find that the level of genetic diversity between urban and rural populations is nearly identical. And there's no evidence of a difference here if you look at the p-value. And there seems to be no relationship between the slope of the decline and the difference in genetic diversity, where genetic diversity was measured genome-wide using neutral fourfold degenerate sites, so putatively neutral sites. So if we go back to the processes that could explain urban and rural clines in cynogenesis, there's no evidence of population bottlenecks, and therefore it's unlikely that neutral processes are explaining the evolution of these clines. Okay, well, what about restricted gene flow? Maybe in combination with natural selection. Maybe in cities where there's a lot of gene flow, we see clines, we don't see clines, but when there's restricted gene flow, we do see clines. So to estimate, get a signature of gene flow. We did this in a number of different ways and something that we're continuing to investigate. But as a first pass, we looked at the strength of genetic differentiation between urban and rural populations using the statistic FST. And what we find is that if you compare the FST between cities without clines in gray or cities with clines in blue and red here, we don't see that cities with clines have higher FST than those without clines. In fact, and if you look at the FST with clines, it's very, very low, 0.011. This is exceptionally low, and it's similar to the FST value without clines. And if we look at the relationship of the slope of the HCN clines with distance from the city center and the degree of FST between urban and rural populations, where each one of these circles is a different city, we see that there's no evidence of a relationship between these. So in looking at the second explanation for the evolution of clines, we see that restricted gene flow is unlikely to explain the evolution of these clines. There seems to be substantial gene flow between urban and rural populations, which is further supported by admixture analyses and coalescent models actually estimating gene flow in both directions. And despite this evidence of substantial gene flow, we still see the repeated evolution of these clines almost half the time. So this leaves natural selection. And at this point, we were quite happy with kind of this result, with the logic that it looks like selection, it smells like selection, therefore it must be selection. When we submitted this paper initially to Science, reviewer number 1 was not completely convinced by our evidence. Reviewer number 1 wrote in their review, "I do not feel that the authors have gathered the analyses to demonstrate the adaptive part. The authors, however, do have, I think, the data to conduct genome scans and/or cline analyses, which would provide a test that the differentiation between urban and non-urban plants in the 2 ACLI loci is stronger than expected compared to the differentiation in the rest of the genome, and hence that these genes are likely under selection in the urban habitat." So, although we were quite convinced, the reviewers were not. And to try and quell this concern and definitively test this, we sequenced an additional over 1,500 genomes for a total of 2,074 genomes. And through the wizardry and know-how of Rob Ness and James Santangelo, we added 2 additional analyses to this study to test whether there is evidence at the genomic level, as well as the phenotypic level, evidence of adaptation by natural selection. The first analysis involved a fairly novel method for genotyping these plants, and I can get into questions later about why it's hard to genotype these plants using genomic data, but we were able to do it through a method that was created by Rob Ness. And he was able to estimate the degree of differentiation at AC and LI and determine the percentage of cities that show FST at one of these 2 loci that is greater than the neutral expectation genome-wide from fourfold degenerate silent sites. And what we found was that in cities that had a significant cline, that the differentiation between AC and LI was greater than the neutral differentiation genome-wide 71% of the time. Whereas for cities that didn't have an HCN cline, it was only the differentiation at ACNLI was only greater than the neutral estimate 33% of the time. And this difference in frequency was at a significant level of 0.067. So quite close and relatively low power given the number of cities that we're talking about here. But this was a genomic test of adaptation that provided what we thought was pretty strong evidence. At the same time, we did a test that was very similar to a QST FST analysis. So we looked at the percentage of cities where the difference in HCN was greater than we would have expected due to neutral processes. How we tested this is quite complicated, and happy to send people the math of how it was done in statistics. But the result boiled down to where there was cities with an HCN cline, 53% of the time, the differentiation in HCN and the phenotype was greater than the neutral expectation. When there was no HCN clines, we never saw the differentiation in HCN being greater than you'd expect by neutral processes. And this difference was highly significant. And so this provides direct evidence for selection on both the genotype and phenotype controlling these clines. So I think this is kind of the best evidence we have right now that this is really, these repeated evolution of these clines is due to evolution by natural selection. We wanted to understand the environmental drivers that were causing this at a global scale. And I won't go through all the analyses because the talk's already a little bit long, but I'll boil it down to telling you the results that changes in vegetation and drought interacted with one another to affect the evolution of clines in hydrogen cyanide. Changes in potential evapotranspiration and changes in vegetation, which we think were actually probably more related to changes in herbivore pressure, both led to evolution of changes in, or were associated with changes in HCN. But there were different drivers in different cities. So in drier cities, it seemed to be potential evapotranspiration that was driving the evolution of this cline, which is consistent with experimental data that shows that there could be direct selection on cyanogenic glycosides as nitrogen transporters in dry conditions to help plants respond to drought. Whereas cities that got a lot more rain, it seemed that there was more changes in vegetation, which is probably related to herbivory pressure that seemed to be associated with these changes in HCN. So if we go back to the original questions, does urbanization lead to convergent environmental change throughout the world? The answer is very clearly yes. Does convergent environmental change cause parallel evolution in an ecologically important trait? Yes, sometimes. What environmental variables drive convergent evolution to cities on a global scale? From the analyses we did, it seems to be vegetation, which is probably related to herbivory and drought. And finally, do other regions of the genome adapt to urban environments in parallel? And I do have the answer to that. I'm not going to have time to go into it, but we have 2,500 genomes and we're mining that for signatures of selection. And the short answer is thousands of regions across the genome seem to be under selection in response to urbanization, which is really quite exciting. So going back to the big question that motivated all of this, do populations adapt to urban environments in similar ways throughout the world? Yes, 47% of the time they seem to be evolving due to evolution by natural selection in response to urbanization. Now, at this point, the audience that's watching this could be asking themselves, okay, this is pretty cool, but is this really important? We're talking about white clover after all. I walk on white clover daily. Well, I would argue that it is, even if you look at this individual system, could be really quite important to ecosystems, especially when you consider that a study published in Nature in 2016 showed that across the entire UK, they quantified the amount of nectar provided by different plants to all pollinators. And the plant species that provided most nectar of all species was white clover. Nationwide, or across the entire United Kingdom, contributed about 30% of the nectar that pollinators acquired from plants. And when you go into urban environments, that number goes to 2/3 of all the nectar that is being provided to pollinators comes from white clover. So the ability for these plants to evolve and adapt to urban environments surely helps them to sustain their populations, which can then have positive feedbacks onto entire ecosystems of pollinators. As well, the plants have a mutualism with bacteria in their roots called rhizobium that extract nitrogen from the air and give nitrogen to the plants, but then it breaks down and also nitrifies the soil. So this could be having very positive ecosystem level effects if these same types of results are playing out in the UK for pollinators as they do throughout the world. So I think this suggests that white clover could be a model for evolution in the Anthropocene, and that's how we've been indeed trying to use it. But we've also think that this study can be show how globally distributed research can increase the scale and impact of our work, not just the science we do, but also by being a model for inclusive collaborative research. Over 50% of the participants and collaborators on our study were women. Over half of the co-authors were early career researchers, graduate students, and postdoctoral scholars. And so I think this has really empowered a lot of individuals to get involved in research at a very high level, at a very large scale. Everything I've told you about is called GLUE 1.0, or what we've affectionately called GLUE 1.0, and we used all of the data generated to launch a second generation of studies that we're calling GLUE 2.0. I don't have time to go into it, but we've launched now 10 new studies, everything from looking at how we can use this system and the data we collected to look at the genomics of invasion and adaptation to climate change, to effects of pollution on cyanogenesis in cities, to how this is influencing natural selection genome-wide. And so this is a pretty exciting next few years for us in the GLUE 2.0 project to understand how urbanization drives evolution of populations using this as a model. But when we span out to the entire globe, I think it's important to think about how not just this one plant, of course, but in the thousands of cities across the world, how are the entire biota that is coexisting within and around cities with us evolving in response to this environmental change that we are so dramatically and rapidly causing throughout the world. I think this is a very important topic and has important applied consequences and may be part of the solution to understanding how to live sustainably on Earth. So with that, I'd like to thank Matthias for organizing the seminar series, and I hope everyone that's watching enjoyed this seminar and learned something about our work. I'll just very quickly thank you for listening, thank the Global Urban Evolution team, as large as they were, for coming along on this voyage with us, and to the very generous funding support that we receive from Natural Sciences and Engineering Research Council, the EWR Stacey Fellowship, the Canadian Research Chairs, and University of Toronto. Thank you.
Wow, thank you very much, Marc. It's been, I mean, what a wealth of information. And I must say, I must confess that I was happy that throughout your talk, as you were presenting, from introduction to the final results, you kept on stealing my thunder, because when you first showed the visible Earth, the first thing I thought was like, wow, I just realized, like you mentioned, having all these urban centers across the globe, this is perhaps the largest experiment on evolution we could ever have, right? So, and then later on you touched upon that. And also I really liked when you uncovered the effects of neutral processes on the evolution of the capacity to produce cyanogenic compounds on white clover. And I have a few questions here.
Sure.
Hopefully they aren't too simple or too dumb. Hopefully I was able
I'm sure not. I'm sure not.
Follow your talk. So eventually you mentioned that, when you were assessing, when you were measuring the power of drift on explaining declines in cyanogenic capacity, you mentioned that epistasis was really an important factor. And the epistatic relationship between those two loci really explains how drift has most times drawn the populations to a non-cyanogenic state. Is that right?
Not exactly. So what we showed was using simulations
Okay.
that population contractions preferentially lead to the loss of cyanogenesis because of the epistatic interaction. So that if cities or any environment are associated with population contractions in this system, it will consistently lead to the loss of cyanogenesis. And as a result, you could get the evolution of declines if you had that demographic process playing out repeatedly in cities where populations were getting smaller in cities. What we found was that's not the case, that the level of genetic diversity in cities and urban environments is really high. And we've since done coalescent modeling, demographic modeling to estimate effective population size in urban and rural environments. This is a paper that's in review right now at Molecular Ecology, led by a postdoc in my lab, Dr. Odd Kazerg. And what she found was that you don't see a consistent change in effective population size in urban and rural environments. And so although it can lead to the evolution of these declines, it does not. It's just a theoretical possibility.
Okay. Well,
that was all based on simulation result, not empirical data.
Okay, that was a good clarification then. And now just to clarify, so all your collaborators that have helped you, sample the white clovers in different cities. So these people were all they grad students, postdocs, or researchers, or are we talking about citizen science, like random people who like the idea and joined you?
Yeah, it wasn't quite community science in that way, or citizen science, as some people call it, in that you had to have some basic level of equipment. The actual sampling was relatively straightforward. We sent people kits in which they could do the assays and collect the plants with and things like that. But you needed to have access to a lab that had pipetters and an incubator in order to do the assay. And that's not something that most people have at home. And we couldn't afford to send out, we send out 180 kits. There was originally 187, 188 cities. And then some people, got injured, not because of the work, or, had to move, or the environmental conditions were unfavorable at the time. And so they weren't able to complete the sampling in their cities. But yeah, so you had to have access to a lab. And that meant that they were scientists that were involved, but at a huge range of career stages.
Absolutely. And, as you described the whole project, I kept just thinking about the management of all of it. Obviously, I do know that you weren't alone in this, but still, I think you must feel very proud of the glue project, because it is, the sheer scope of it is monumental.
Yeah, we feel pretty good about it.
Of course.
I do want to say that this was not a sole effort by far. We had a lead team of 12 people. So, the three main people that were kind of at the leading edge of that lead team were Rob Ness and James Santangelo. James Santangelo was a PhD student between our labs who ended up being the first author on the paper, shared with me. But then there was a series of technicians that were absolutely vital to the project. There was current and past graduate students that were helping to innovate techniques to figure out how could we ship tissue for whole genome sequencing. How do we make these assays so that they're foolproof? And we learned a lot along the way, and learned from our own mistakes. And even when we thought we had it just absolutely perfect and that no one could interpret it in different ways, then we'd find out, oh, apparently they can.
Yeah. So that's how science progresses,
But it
right?
all came together with a lot of work, and we had at the end when it came out in Science, we had a big party online because it was
Oh,
during COVID.
nice. Yeah.
So we had a big Zoom party with people from Australia and Japan and Europe and South America. It was fabulous.
Oh,
So really fun.
I'm happy to hear that. And and I must say that again, the main goal of this series, of our online series, really is to promote studies on evolution and hopefully to get more people attracted to the topic
Yep.
of evolution as a whole. And what I found very interesting about your glue project is that it brings closer to people's day-to-day lives, right? Because I think when people hear about evolution, they either think about, that misleading illustration of primates evolving towards humans, right?
Right.
Or perhaps if they have, if they have some knowledge on biology, they might think of Drosophila, for instance, or allele frequencies in populations. But all those examples are really far away from their everyday lives. And then you're bringing this idea that evolution is happening around us, especially if you're living in urban centers. And I think we are going to be seeing more and more studies on that arena. As you beautifully said, we are now in the Anthropocene. I know there is still some controversy within the geologists, but still, I mean, I think the ecological and social meanings of the term Anthropocene are clear, crystal clear to us, and it really showed that we should be paying attention to it. And I mean, the studies were on white clover, but I'm here thinking about even other species and all the possible implications of it. So yeah, thank you very much. This was very insightful, and I'm pretty sure that viewers will appreciate your time to promote all those results.
No, thank you. I appreciate that, and I've included a link in the chat that hopefully you can share with
Mhm.
viewers that shows this was one of the Glue 2.0 projects that was shows an animation of the project and the results, which I think really helps to enhance and broaden the understanding for the public about how you and I and everyone that's watching here is altering evolution around us every day in
Yes.
literally our own backyards. And so, yeah, I hope they enjoy the animation. It's very short, but I think it really captures well what we've done here and what we found.
Yeah, absolutely. I'll attach to our recording. Well, again, thank you, Mark. I will stop the recording now, and then
Okay.
we can chat for a few minutes as well. Okay. Thanks
So
again.